Now, the title of today's presentation is Introduction to Microbiology. This presentation is targeted at the beginners in microbiology. It is aimed uh, to assist students to be able to recall what has been discussed during the normal classroom lectures. Now, to start with, what is microbiology? How do we define microbiology? Microbiology is generally a branch of science that deals with the study of living organisms that are individually too small to be seen with unaided eye. It's a branch of science that deals with the study of living things which are individually too small to be seen with our own natural eyes. Hence, these living organisms are called microorganisms. They can only be seen with the aid of microscopes or other magnifying devices, which magnify objects. So in general term, microbiology, the science of microbiology deals with the study of microorganisms and their activities. Now, there are various types of microorganisms, but for simplicity, they can be divided into two major categories: the acellular microorganisms and cellular microorganisms based on their levels of organization. The acellular microorganisms are generally the viruses, which are not up to a cell level of organization. They are generally microorganisms that have the simplest structure and the smallest body size, which is only made up of uh, an outer cord and a nuclear material. So it's just made up of a nuclear material surrounded by an outer proteinous cord. Because of their smaller nature, viruses are capable of free living. That is to say, viruses cannot survive on their own. They depend on other living organisms in order to reproduce and carry out most of their body activities. Hence, viruses are generally defined as the smallest obligate intracellular infectious particles that are made up of nucleic acid plus surrounded by protein coat plus or minus an outer cover, which is called envelope. Now, regarding the cellular organisms, the cellular organisms are categorized into two. The first category are the prokaryotes. Prokaryotes, prokaryotes are microorganisms that are a little bit more advanced than the acellular microorganisms or the viruses. They are living organisms that have cellular organelles which are capable of supporting them to live freely in the environment. However, they lack most of the membrane-bound organelles, and hence they are referred to as the prokaryotes, living things with incomplete cellular organelles, cellular structures. Now, this category of microorganisms are generally made up of bacteria family. They are generally bacteria. We have the true bacteria, we have the extreme bacteria, archaea, we have mycoplasma, which are bacteria that lack uh, cell wall. We have the smallest group of bacteria, Rickettsia and Chlamydia, which we are going to see during the course of this discussion. So, in essence, the prokaryotes are made up of the bacteria family. Now, the second division of the cellular microorganisms are the eukaryotes. As the name implies, eukaryotes are defined as living things that have complete cellular organelles. They have complete cell membrane bound organelles. They have complete cellular organelles. Now, in this category, we have 4 groups of microorganisms under this category belonging to, we have fungi belonging to kingdom fungi, we have protozoa belonging to kingdom protista, we have algae, we have helminths belonging to kingdom Animalia. Now, altogether, if we add everything together, from the acellular part, we have viruses. In the prokaryotes, we have 5 categories of bacteria, and then under the eukaryotes, we have 4 categories: fungi, which include both unicellular and multicellular types, and we have protozoa, which are only unicellular, and algae, which are unicellular and some multicellular, and then we have the helminths, which are all multicellular. So that is to say, we can categorically say that microorganisms are of 3 different kinds: the acellular, the prokaryotes, and the eukaryotes. And altogether, we have about 10 groups. We have viruses, bacteria, archaea, mycoplasma, rickettsia, and chlamydia. And then we have fungi, protozoa, algae, and helminths. Now, having known the definition of microbiology and microorganisms, let us quickly look at the scope of microbiology. The scope of microbiology simply means the area of coverage of the discipline of microbiology. What microbiology covers? What are the aspects covered by microbiology? What are the important aspects? What are the important branches of microbiology? Now, based on the scope, uh, uh, microbiology generally study microbial forms, structure, reproduction, physiology, genetics, metabolism, habitat, and taxonomy of microorganisms, with particular emphasis on the beneficial and detrimental effects on peoples and other living things, as well as the physical and chemical changes that microorganisms cause in various substances. That is to say, microbiology studies the general aspect of microorganisms and with emphasis on their beneficial and detrimental effect, as well as the physical and chemical changes that microorganisms can cause on various substances. Now, based on this general scope, uh, we can simply say that microbiology uh, is divided into the study of the basic aspect of microorganisms and the study of the applications of the microbial knowledge for the benefit of human kind. Therefore, microbiology, based on this, is divided into two. We have basic microbiology and we have applied microbiology. Now, the basic microbiology studies the uh, basic aspect of microorganisms generally, such as the types of microorganisms, branches based on types of microorganisms, based on the habitat, and taxonomy, physiology, reproduction, genetics, everything about uh, the basic aspect of microbiology. Everything about microorganisms comprises the uh, the general knowledge of microorganisms. The comprises the basic aspect of microbiology, and it is subdivided into three major subdivisions. That is to say, the branch of basic microbiology has three sub-branches. The first one is a subdivision which is based on the types of microorganisms under study. Remember, we mentioned various types of microorganisms. So the first subdivision of basic microbiology uh, comprises all branches of microbiology that specifically study the type of microorganism. That is, those that specifically specify or specialize in a particular type of microorganisms. For example, we have bacteriology, which is the study of bacteria, and the scientists that study bacteriology are called bacteriologists. Similarly, we have mycology, which is the study of fungi, and the scientists that study fungi are called mycologists. We have protozoology, study of protozoa, and the scientists are called protozoologists. We have virology, study of viruses, and the scientists studying virology are called virologists. We have pycology or algology, which is the study of algae. We have chlamydiology, we have rickettsiology, and so on. Just as the number and the type of just based on the types of microorganisms. Now, the next subdivisions of basic microbiology is based on the functional processes of microorganisms being studied. This time, uh, uh, it's not specific on the type of microorganism, but based on the general functional processes taking place in the body of microorganisms. This aspect include like uh, microbial physiology, which is a branch of microbiology that deals with the study of the functions of microbial cell organelles. We have like microbial genetics, which is the study of genetic changes in microorganisms and how they are manipulated in recombinant DNA technology for the production of desirable products. We have microbial metabolism, um, which studies the all metabolic aspects of microorganisms. We have microbial reproduction that studies reproduction in microorganisms, microbial morphology, which studies the shapes and structures of microorganisms, microbial cytology, which is a branch of microbiology that studies microbial cells, and so on. Now we have the last sub-branch of basic microbiology, which is based on the habitat of microorganisms. That is the branches of microbiology based on habitat of microorganisms. Like we have the land or soil habitat, and the branch of microbiology that deals with the study of microorganisms on land or soil habitat is called soil microbiology. We have marine microbiology, which is a branch of microbiology that studies microorganisms in the oceans. We have freshwater microbiology, which is a branch of microbiology that studies microorganisms in freshwater bodies and their activities. We have air microbiology or aeromicrobiology, which is the branch of microbiology that studies microorganisms in the air and microorganisms and airborne disease microorganisms. Now, coming to the applied microbiology. In the applied microbiology, the branches here are based on the application of knowledge of microorganisms for the benefit of mankind. So these are specialized subdivisions of microbiology which studies microorganisms based on how they are applied, how their knowledge is applied for the benefit of mankind. Like we have pharmaceutical microbiology. Pharmaceutical microbiology is a branch of applied microbiology that involves the study of microorganisms associated with the manufacture of pharmaceuticals, research and development of anti-infective agents, antibiotics, the use of microorganisms to detect mutagenic and carcinogenic activity in prospective drugs, and the use of microorganisms in the manufacture of pharmaceutical products such as insulin, human growth hormone, and so on. Next, we have agricultural microbiology. This is a branch of microbiology that studies microorganisms in relation to agriculture. It includes soil and plant microbiology, and it deals with plant associated microorganisms, soil associated microorganisms, and plant associated microorganisms, plant and animal diseases, and so on. So this particular branch of microbiology deals with all aspects of agriculture, including microbiology of soil fertility, such as microbial degradation of organic matter and soil nutrient transformation, etc. Now, the next one is food and dairy microbiology. This particular branch of microbiology deals with the study of microorganisms involved in food processing, development, spoilage, as well as hazards associated with food. We also have environmental microbiology. which studies the composition and physiology of microbial communities in the environment. The environment in this case refers to the soil, water, air, and sediment covering the planet, and can also include animals and plants that inhabit these areas. So environmental microbiology include the study of microorganisms also that exist in artificial environments such as bioreactors and so on. Now, another important branch is public health, microbiology, and epidemiology. This is a specialized branch of applied microbiology concerned with the mechanism of infectious diseases, with an emphasis on microbial pathogens, uses and application of modern biotechnologies, and epidemiologic skills relevant to the prevention and control of problems in the community arising from infectious diseases. We also have immunology, which is the study which studies all aspects of immune system, including the structure and function, disorders of immune system, blood banking, immunization, and organ transplantation in relation to microorganisms. We have another important branch, which is microbial biotechnology, which deals with the manipulation through genetic engineering of microorganisms or their component to produce useful, usually commercial products, uh, such as like pest-resistant crops, new bacterial strains, novel pharmaceuticals, uh, and also any of the various application of biological science used in such manipulations. We have genetic engineering and recombinant DNA technology. In this branch, it deals with a set of techniques that allow the identification, manipulation, and multiplication of genes of microorganisms in order to make them produce improved varieties or products. Uh, we also have industrial microbiology, which is a branch of applied microbiology that study microorganisms used to produce industrial products such as alcohol, organic acids, amino acid, vitamins, etc. We have geomicrobiology, which studies microorganisms in relation to geology. We have pathogenic clinical microbiology, which studies microorganisms that cause diseases in humans, animals, and plants. So all these are branches of applied microbiology. That is specializations in which microbial knowledge is applied. So these are the this this entails the scope or area of coverage of microbiology, the basic and applied microbiology. Now, now, why do we study microbiology? What is the importance of microbiology to us? What benefit is microbiology offering to us? In essence, what is the relevance of microbiology to our life? To answer this question, it takes us to the next aspect of the discussion, which is relevance of microbiology. Relevance of microbiology simply we can link it to the influence of microbiology or microorganisms to human beings and other organisms, as well as how microorganisms are manipulated to serve for specific purposes.
That is to say, study of microbiology help us to understand the basic aspect of microorganisms and how we can apply the knowledge of microorganisms for the benefit of mankind. Uh, uh, One of such roles of microorganisms, which we understand through the knowledge of microbiology, is the general role of microorganisms in environmental and ecosystem balance. Microorganisms carried out natural role in the environment, making our environment and ecosystem balance through a number of processes such as decomposition of organic matter to break down breaking down of waste materials and dead organisms, making our environment clean. Uh, Microorganisms also carry out nitrification, that is fixation of nitrogen and making it available to the plant in form of nitrate. Example is nitrogen fixing bacteria. Also, through the decomposition process, many microorganisms are involved in nutrient cycles, cycling essential elements in nature, such as the carbon cycle, nitrogen cycle, oxygen cycle. All these cycles requires microorganisms to complete. Now, in addition to decomposition, microorganisms also carry out another important natural role, which is photosynthesis, where they serve as producers in most food chains. Example of such microorganisms are the photoautotropic bacteria and algae, uh, both of which serves to photosynthesize and serve as producers in most food chains, especially in aquatic environment. Now, in addition to the larger environment, microorganisms also serve many important roles in microenvironment, such as in the digestive system of humans and animals, where we have some microorganisms like Escherichia coli, Bacillus pyrrhocyanus, etc., that convert undigested and undigestible foods in the colon to feces, and in the process, they produce some essential vitamins and other minerals that are absorbed and used by the body of the host. Now, apart from environmental and ecosystem balance, uh, the study of microbiology help us to understand some of the traditional applications of microorganisms. Traditionally, human beings have been using microorganisms for 1,000 years, and it is with the knowledge of microbiology that we come to understand such applications. For example, uh, bakers and brewers yeast, which are used in baking and brewing process, cheese production. In fact, some microorganisms are even used traditionally to kill germs. For example, moldy breads are applied on wounds traditionally to kill germs. Now, the knowledge of microbiology also help us to now apply the knowledge, modern application of microbiology, to apply the microbiological knowledge for certain benefits, like in industrial and food microbiology application. Study of microorganisms help us to be able to apply microorganisms in food and industries uh, to produce important products. For example, in making and preserving food products such as yogurt, salami, cheese, and so on. In the production of important compounds such as antibiotics, monosodium glutamate, amino acids, sweeteners such as aspartames, uh, uh, solvents like ethanol, propanol, and so on. Another area of modern application of microorganisms is in biotechnology. In biotechnology, human beings manipulate microorganisms to make products in industrial settings. Example of such areas is in genetic engineering, where microorganisms are manipulated to create new products. For example, genetically modified organisms such as crops that are resistant to weeds, resistant to insects, and so on. Another biotechnological application is in recombinant DNA technology, which apply microorganisms to be engineered to synthesize desirable products such as drugs, hormones, enzymes, vitamins, and so many others. Another important area of microbiological application is in petroleum industries. In petroleum industries, microorganisms are used for a number of processes. For example, some microorganisms are used to produce fuels. Microorganisms like methanogens are used to produce biogas, which is used for cooking and generating electricity and so on. In addition, microorganisms are also used in processes like in secondary oil recovery, because most microorganisms crude oil formation start with microbiological activities. So some microorganisms are used, applied for oil recovery in petroleum industries. Another application is in bioremediation. Many microorganisms are used to ameliorate crude oil contaminated environment as a result of accidental oil spillage in the environment or other spillage processes which makes the soil infertile. So microorganisms are applied to make the soil fertile again. Example of such microorganism is Candida paraphinica, which is a fungus that is capable of growing in crude oil contaminated environment to break down the hydrocarbons, volatilize them, and at the same time it produces large biomass which can be harvested and used as fuel. The biomass can be harvested and used as food. Now, another modern application is in agriculture, agricultural microbiology, in which microorganisms are used for a number of processes such as transmission of desirable traits between plants. Some microorganisms can be used to transmit genetic characters from one plant to another. Example, Agrobacterium tumefaciens is used to transmit genetic characters from one plant to another. Some microorganisms are also used as bioinsecticides. For example, some fungi such as Leptolegnia caudati and Apomyces laevis have are capable of parasitizing on mosquito larvae and are therefore used in the control of such insects. Another example is Bacillus thuringiensis, which is a bacteria that parasitizes on insect pests, and it is therefore applied on field in the farms to control pests of crops. In addition, in agriculture, microorganisms are also used to increase soil fertility through biofertilizer production. Some microorganisms are added to the soil directly or are used to produce biofertilizer to make the soil fertile. Example of such microorganisms Pseudomonas species, blue-green algae, and so on. In fact, research has shown that some microorganisms such as nitrogen utilizing species like blue-green algae, when applied in polluted soil, were found to increase crop yield significantly. One other area in which microbiology is applied is in the field of geology. In geology, microorganisms have been used in biohydrometallurgy for extraction of metals from their ores, a process called bioleaching and biomining. For example, Acidocyanobacillus peroxidans, Acidocyanobacillus cyanoxidans are bacteria that are used in bioleaching of common heavy metals such as copper, zinc, nickel, lead, arsenic, cobalt, molybdenum, gold, and silver. On the other hand, some fungi such as Aspergillus niger, Penicillium simplicissimum are fungi that are used in bioleaching of metals such as copper, tin, aluminium, nickel, lead, and zinc. Now, the science of microbiology is also relevant in understanding the negative influences of microorganisms. Through the study of microbiology, we come to understand negative influences of microorganisms and how or the strategies we can apply to reduce such effects. Some of these negative influences of microorganisms include disease causation, that is, animal and human diseases, including allergies as well as plant diseases. Many microorganisms are responsible for such diseases. Now, the study of microbiology help us to understand the disease causation process and how to prevent such diseases. In addition, microorganisms cause food infection. Food infection is another negative effect of microorganisms which makes food unsuitable for eating. In addition to food infection, some microorganisms cause food spoilage. They spoil the food completely. For example, several agricultural products such as vegetable cereals as well as processed foods are being spoiled by microorganisms through the process of fermentation, rancidity, or putrefaction process. In addition to food, some microorganisms also cause food intoxication. They produce certain poisons in foods. They produce toxic substances which are capable of causing diseases. For example, mycotoxins produced by fungi which causes mycotoxicosis, and staphylococcal food poisoning, and so on. Another negative influence is damage of valuable materials. Many microorganisms through decomposition process cause damage of valuable materials such as food, lumber, paper, clothing, magnetic tapes, disc, glass lenses, marble statues, bones, wax, etc. In fact, the science of microbiology help us to understand the negative influences of microorganisms and sometimes even how to utilize such negative influences for our own use. For example, humans have been using microorganisms to produce biological weapons, biological bombs that are used for mass destruction, in bioterrorism, and so on, to kill millions of peoples uh, uh, within short period of time. So, in fact, microbiology has great relevance to human beings because microorganisms have been used to produce variety of desirable products that are used either in industries, agriculture, and so on, as we have seen. Let's look at some additional examples of products that are produced from microorganisms. For example, many microorganisms have been used to produce foods and beverages in industries such as teas, alcoholic beverages, which are produced by bacteria or yeast, by fermentation of sugars in fruit juices. We have products like soy sauce, which are produced by fungal fermentation of soybeans. We have products like vinegar, which are produced through bacterial fermentation of sugar. Yogurt also produced through bacterial fermentation of milk. Soy cream also produced through bacteria. We have artificial sweeteners which are commonly used in preparation of drinks, sweetener like aspartame are produced by microorganisms in form of which amino acids synthesized by bacteria from sugars. We have bread making also involves the use of microorganisms. Other products such as antibiotics are produced by bacteria and fungi. Microorganisms are also used to produce hormones like the human growth hormone, insulin, and so on. Some microorganisms are also used to produce laundry enzymes, vitamins, drain opener, and so many important products. So microbiology has great relevance to our lives, and hence the study of microbiology is very important. Now, having learned so much on the definition, scope of microbiology, relevance of microbiology, the next question we may ask ourselves is, how does the microbiology started as a discipline? When was the study of microbiology started? Who are the scientists that contribute to the development of microbiology as a discipline? So this takes us to the next aspect of this presentation, which is brief history of microbiology. So microbiology, just like all other science disciplines, was developed through series of stages. In fact, microbiology as a discipline started when microorganisms were discovered, and this follows some series of stages. Stage number one, which can be referred to as the ancient belief. During the ancient period, many people do not believe in the existence of microorganisms, and therefore their major belief regarding diseases is that diseases were believed to be supernatural in origin. Diseases were believed to be a punishment sent from God for the sins of humankind, and therefore during that period, people make a lot of sacrifices, lustrations to appease the anger of gods in order to prevent diseases from happening. Now, the ancient belief was followed by the period of intervention. During the period of intervention, which we can call the second stage in the development of microbiology, is that at this period, human intervention started. Some scientists began to think that diseases are not just punishment from God, but they are as a result of certain natural processes. And the first person that started making contribution towards this is the Hippocrates, who is now considered the father of medicine. Hippocrates introduced the concept of humoral theory of diseases in around 350 BC. He stated that diseases are caused by imbalance of body fluids. That is humoral theory. Because of this, Hippocrates introduces certain traditional practices like bloodletting and causing individuals to vomit in order to treat some diseases, common diseases. However, humoral theory does not methods does not completely treat all diseases, and therefore, so many other people began to think on how to treat diseases. So, after humoral theory was followed by the herbal healing practice, which was first introduced by Asclepius, who is now regarded as the god of medicine. So Asclepius started using some herbs to treat people, together with his two daughters, Hygeia, who is now considered as the goddess of health, and Panacea, that is now considered as the goddess of pharmacy. So together, they also introduce the concept of contagion, which states that diseases can be spread from person to person through direct or indirect body contact. Hence, sick people are always isolated from healthy persons during that period. Now, as if the knowledge of microbiology will progress very well, but then one important thing happened, which is a theory of spontaneous generation, or abiogenesis theory, introduced by Aristotle in around 400 BC, which prevent further progress in investigations toward discovery of microorganisms. The theory of spontaneous generation stated that living things develop spontaneously from non-living matter that is left standing. So Aristotle advocated this, and many people believe in this idea until it was challenged by one of the Italian physicians, Francisco Redi, who proves that goose placed over jar containing meat prevent maggot from developing from the meat product. So similar experiment by other scientists help to discredit the theory for larger organisms. So in general, there were three major factors that retard the development of microbiology. The first one is the concept of abiogenesis introduced by Aristotle, and the second aspect is that there was lack of magnifying devices to view microorganisms. And number three, there is also lack of improved laboratory techniques that can be used for investigation of microorganisms. So these are uh, some of the factors that retard the development of microbiology uh, process. Uh, Now, the next stage is the era of discovery of microorganisms. Uh, Discovery of microorganisms started with the work of some investigators, such as the Roman philosopher Lucretius, about 100 BC, and physician scientist Girolamo Fracastro, uh, who suggested that diseases are caused by invisible living creatures, invisible uh, living things, invisible small animals in the air, uh, uh, called germs or seminaria or seed as they describe them. However, not much progress was made until one of the first magnifying devices was made by Robert Hooke in 1665, uh, who first observed bacteria and protozoa with his invented microscope and drew their structure in his book, which he called Micrographia. But unfortunately, uh, Robert Hooke did not make accurate drawing of this agent, and that is why he was not given the full credit of discovery of microorganisms. So the real discovery of microorganisms, real description of microorganisms was started by the Dutchman Antony van Leeuwenhoek in around 1673, who gave the most accurate description of bacteria, various shapes of he described various shapes of bacteria, such as the spherical shape, the so, because Antony van Leeuwenhoek was the first scientist that accurately described bacterial shapes, uh, uh, he was later referred to as the father of bacteriology. In around 1878, another scientist, Sedlot, introduced the term microbes to referring to microorganisms, and which is now replaced by the word microorganisms. Now, immediately after the discovery of microorganisms by Antony van Leeuwenhoek, scientists become more interested in microorganisms, and that led to the next stage in the development of microbiology, which is a period of scientific development of microbiology. And during this period, the theory of spontaneous generation was completely discredited by the work of some scientists, such as the Italian priest uh, Lazzaro Spallanzani, who is also a naturalist. Uh, he boiled beef broth for an hour, sealed the flask, and observed no formation of microorganisms. In addition, another important scientist, Louis Pasteur, in 1859, in public, he prepared boiled broth in flask with long, narrow gill neck tubes that were open to the air. So air can pass, but microorganisms settled in the gooseneck, and with that, no growth developed in the flask. So if the necks were broken, growth commenced immediately. So with this experiment, Pasteur had not only resolved the controversy over spontaneous generation, but also has shown how to keep a solution sterile through the boiling process, which is now referred to as the pasteurization process. Now, in addition to the Louis Pasteur's contribution, another scientist who is an English physician, John Tyndall, proves that dust did not indeed carry germs. So he dealt a final blow to the spontaneous generation in 1877, where he introduced the Tyndallization process. So John Tyndall explained that dust indeed carry germs, and that if dust was absent, nutrient broths remain sterile, even if directly exposed to air. So he was able to also explain satisfactorily the need for prolonged heating to eliminate microbial life from infusions, which is a process that is referred to as Tyndallization. So after this discovery, the development of microbiology progresses. Microbiology continued to progress rapidly with the contribution of various scientists, some of which include, for instance, the Louis Pasteur. So Louis Pasteur made great contributions to the field of microbiology, as a result of which he is commonly referred to as the father of microbiology because of his numerous contributions, such as he was the first person that coined the term microbiology. He discredited the theory of spontaneous generation. He developed techniques for cultivation of microorganisms. He proposed the germ theory of diseases, which states that diseases are caused by indivisible organisms, which are caused by individual organisms, which are called germs. He also introduced pasteurization technique of killing microorganisms through boiling process. He also discovered attenuation process and so many other contributions which led to the continuous development of microbiology as a discipline. So microbiology continued to progress with the contributions of so many scientists. We have another important scientist that contribute tremendously, that is Robert Koch. Robert Koch made a lot of contributions to the development of microbiology. He was the first scientist that discovered the causal agent of anthrax in 1876. He also discovered the causative agent of tuberculosis and cholera in 1882 and 1883 respectively. He also introduced the staining techniques, which is commonly used for microscopic observation of microorganisms in 1877. In addition, he explores the use of animal experimentation in establishing the cause of diseases, and with that he developed what is now called as the Koch postulate of diseases. Robert Koch observes that in any case of diseases, microorganisms can be isolated from the uh, host, from the infected host. Microorganisms can be isolated, and those microorganisms responsible for the disease, if injected into an experimental host, the same disease conditions happen, and the same microorganisms can be re-isolated from that infected host. So with this, he introduces what is now called as the Koch postulate of diseases, which states that in all observed cases of a given disease, microorganisms must be found in pathological relationship to the symptoms and lesions. And then number two, those microorganisms can be isolated from the victim. And number three, the same microorganism, when inoculated into susceptible hosts, must reproduce the disease symptoms originally noted. And number four, the same microorganism must again be isolated from the experimental host. In addition to these contributions, Robert Koch also introduced the method for isolating few cultures of bacteria in 1881. So Robert Koch made so many contributions. He was the first that observed hypersensitivity reaction for second encounter of diseases in pigs in around 1890. He also invented hot air oven and steam oven. So in addition to Robert Koch, we have many other scientists that contribute to the development of microbiology, some of which include Joseph Lister, who is now considered as the father of modern surgery because of his contribution to the modern surgery. He was a scientist that developed antiseptic techniques for destroying microorganisms, and he developed a system of antiseptic surgery designed to prevent microorganisms from entering into wounds in around 1867. Another scientist, Edward Jenner, who is now referred to as the father of immunology, introduces the modern method of vaccination in around 1796. Now, in addition, we have another scientist, such Paul Ehrlich, who is a German scientist and a father of modern chemotherapy. He's regarded as the father of modern chemotherapy because of his numerous contributions to to the chemotherapy. He initiated histological stains of cells and tissues to reveal their composition. He reported the acid fastness of tubercle bacilli. He introduced the method of standardizing toxins and antitoxins, and coined the term minimum lethal dose. He also proposed side chain theory of antibody production, and he created a new branch. With this, he created a new branch of medicine known as the chemotherapy. So this is a brief history of microbiology, which progresses slowly and continue with the discoveries made by several scientists.